Alright guys, so this video is about the biochemistry and it's the DNA replication, and I spent much of my morning relearning this stuff, so had to go for a drive. And anyways, it's all about telling that story, and I think if you can have that foundation, I think with the more questions that you see on this topic, I think the better off you'll be. So anyways guys, learn to tell that story, and hope you find the video helpful. Alright guys, so this question is going to read, a diagram explaining DNA replication is shown below. Which of the following would be representative of the function of ligase? Alright, so this is the DNA replication, but before we do this, let's go ahead and kind of explain how this process works. And I think, because then we can come back and we can do questions like this actually rather easily. So DNA replication, okay? DNA replication is what we're going to be talking about. And of course, you're going to tell a story. Now, the key with this whole deal is I need you to know and understand DNA, even RNA, synthesized in the 5' to 3' direction. Okay, very important. I mean, that's just base knowledge, and I've seen even questions on NBME that just ask as simple as that, 5' to 3'. So lock that in. Now, some other little things that you'll see in that Usmle, your book, and they'll talk about how DNA is very tightly wound up or winded or whatever you want to say, just so it's packed nice and neatly. And so you'll see these things, and you'll see a diagram like this before. And remember how they said like the DNA is wound, the one that's not in the nucleosome core, the one that's not in, is going to be H1. Okay, and then the other ones, the H2A, H2B, H3, H4, we don't care. Those are in the nucleosome core. So I've seen questions about making sure the DNA, knowing which one's not in the nucleosome core is H1. And what do we care about these things, right? What's some other terminology that we got to be familiar with is when things are methylated, things like that. When they're methylated, they are muted, okay, methylated, muted, and then the word acetylation activates, okay, activates. But you can also think like when something is not active, it's tightly bound, okay, tightly bound, and then if something is going to be more active, it's loosely bound. When it comes to DNA, when it comes to this kind of stuff, tightly bound is inactive, loosely bound is going to be active. Okay? And then some other things, some simple things are going to be like, what was our start codon? Okay, our start codon is going to be Mr. AUG, and then we talked about him, right? We talked about him on the video talking about homocystinuria and stuff like that, because remember, homocysteine takes a left turn, B12, etc., goes to methionine, AUG, start codon. And then the stop codons, okay, the stop codons are what? UGA, UAA, and UAG. You just can't get through any type of biochemistry review without knowing the stop codons. And of course, in step 1 or any book, really, it'll say, you go away, you are gone. And again, these are all stop codons. So, back to this whole DNA thing, DNA replication. So, normally I'm going to say, things that's kind of packed in there tightly, and I'm going to do my best job trying to draw this here. But, DNA is packed pretty tight, and they have to identify on a question, really, where is the 5' versus the 3'? So they got to tell you that. So to replicate DNA, we have to kind of, we nick it, because that identifies where we're going to start, and then it gets pulled apart, okay? Now, before we draw into that how it gets pulled apart, there's a couple terminologies that I want to write down in the beginning that we're going to identify as we go. And those are going to be topoisomerase, helicase, single-stranded binding proteins. Let me see, let's go RNA primase. Okay, let's say RNA primase, or RNA primer, I should say. Ligase, DNA-dependent RNA polymerase, Okazaki, right? Okazaki fragments. And then we're going to have polymerase 1, and it's a DNA polymerase. And then polymerase 3. Okay. Now, at the end of this little question thing here, we're going to know how to each one of these. And if you know each one of these and where they placed and what they do, really, when it comes to biochemistry, there's nothing they're going to ask you when it comes to replication that you're not going to, you're not going to have any difficulty with. So then let's draw basically when it replicates. And so all I did was take, say, this section right here, and now I'm just going to expand on it a little bit. Okay, I'm just going to expand it. So anyways, again, I got to have this identified. There's my 3', 5'. So down here is because it matches, right? So there's my 5' and my 3' ending. So if I separate this guy, I got to determine, okay, which way it's like a zipper, right? And which way am I going? I got to tell myself which way am I making this. And so I'm going to make it this way, okay? And so this guy that separates this guy, kind of puts it apart, is who? He is going to be known as helicase, right? And, you know, helicase kind of separates it. But why I mentioned of knowing that we're going in this direction is because as we're making this DNA, now here's the codons per se, you know, on the parent strand, because what we're going to do is make new strands, or they call them the daughter strands. And so we know that if we're going this way toward where we kind of, you know, are separating it, then we're moving towards, right? We're moving this guy, the strand right here is moving towards the where we separated it, or the replication fork is what is how they call it. Because remember what we said back up top, everything's made in the 5' to 3' direction. So it's not like we can go on this bottom strand and go 5' to 3' going this way, right? Well, how come, right? How come I can't just do this? Because in theory, if the 5's here, it means that the daughter strand's going to start with would start with the 3', and you have to go to the 5. And when you're like, well, wait a second, that's not what the rule is. The rule says it's made 5' to 3'. So therefore, this is where the questions come in on the test. You have to know, again, on the top strand, or the leading strand, we call it the leading strand because it moves towards the area of separation, or towards the replication fork. You know, it's made from the 5' to 3' direction. And that's cool, it's great. You know, it's nice and continuous, okay? Now, this bottom strand, since we can't just go in this direction because that would be from 3' to 5', which goes against the rules, what it does down here is it makes fragmented or smaller pieces. So it would say start here and go that way, right? Because that's holding the integrity of going from a 5' to a 3' direction. It would start here and go 5' to a 3' direction. And again, here, 5' to a 3' direction, holding true the rules. Okay? This is how they're going to test you on this. Now, what started this stuff, right? What started it? Well, I got to tell you that, you know, if I were to take a small step back, that the energy, you know, if I was really to dig in deep, and you can see this on your step books, is, you know, I'm trying to think how to properly say this, is, anyways, the energy, the 5' end, I say this is the 5', this is 3' end, the 5' end of something coming in or towards this, okay, this is like a codon per se, the nucleotide, is that it uses the 5' of the incoming nucleotide binding energy, and the 3' of, you know, of the target is where it gets it. So okay, I'm not doing a very good job on that. But anyways, long story short, they need the energy from this connection of these two guys. but it's the 3' end to the 5' of the incoming. The 5' incoming goes to the target of the 3'. Anyways, it uses that hydroxyl group because this hydroxyl group provides the energy. Okay, provides the energy. Because who's going to start this stuff? Like, even though I started here and go into the 3', I needed somebody to start that. And how that works is, it's what you need to know is it's going to be his name. It's going to be the RNA primer. Okay, RNA primer. And he's going to be there, and I'm going to have him down here. You know, anywhere where I started, I'm going to call him the RNA primer. RNA primer. But again, he requires, and well, I should say this, he sends DNA polymerase 3 to do the work. That's how I tell the story. So DNA polymerase 3, he goes out and does the work, meaning he lays the track on this new strand. So I got an RNA primer here. He's going to say, Mr. DNA polymerase 3, I want you to go on your way and lay the track all the way down, okay? Oh, and by the way, when you do that, make sure you proofread it, make sure everything's okay. Again, so RNA primer tells DNA polymerase, go ahead and go that direction. And again, it's just for the simplicity of the story. RNA primer here is going to tell DNA polymerase 3, again, go that way. And again, on your way down there, Mr. DNA polymerase, I want you to make sure you check and make sure, read everything, make sure it's correct. Now, this RNA primer, we can call him primase for the most part. Primase technically forms the RNA primer through, okay, now, how can I say this? The enzyme primase, which is the primase, which kind of fuels the primer, is DNA dependent RNA polymerase. Okay, so basically primase, which gets its energy from DNA dependent RNA polymerase, forms the RNA primer. Okay, so again, we talked about energy from the hydroxyl group. The 3-hydroxyl group provided by the enzyme primase, which is DNA-dependent RNA polymerase, forms the RNA primer. Okay, that's probably the best way to say it. Again, let me just write it down right here. The hydroxyl group provided by primase, okay, and we can just put primase here, kind of right next to RNA primer because that's who he makes. And this primase, which is what? DNA-dependent RNA polymerase, forms RNA primer. And again, RNA primer is at the beginning of the 5' and tells Mr. DNA polymerase 3, go on your way. I'll stay right here. You go on your way and lay down the track. He does it up top in the leading strand. And again, he's a leading strand because he's moving towards the replication fork, 5' to 3' direction. And in the lagging strand, he makes these smaller segments. But again, they start here and go this direction. They're moving away. Whoops, I'm sorry. They're moving away from the replication fork. But again, the primase is going to make the RNA primer, but the primase, he has the hydroxyl group and he's DNA-dependent RNA polymerase, makes the RNA primer, which tells DNA polymerase 3, you go that direction, you go in 5' to 3' direction. Oh, and by the way, I want you to proofread everything along that direction as well. Now, these little sections right here, these small fragments, okay, these small fragments are called what? Akasaki fragments. Akasaki fragments. Okay, so let's go back and look at these this terminology for us real quick. And actually, I didn't get into this, but originally, when we were up here and we said, well, where do we start this? Where do we start the nick or whatever? That's the job of topoisomerase. Topoisomerase is going to nick the DNA and say, this is where we're gonna, this is where we're gonna have our, have our party. This is where it's going to start. So topoisomerase, you can say, kind of nicks it, and then it's going to stabilize for the most part. It's going to stay ahead of the helicase. It's going to stay there, and the key word with the topio is they call it negative supercoiling as opposed to positive supercoiling. Now, supercoiling means I want to kind of collapse this thing back to this super where did I draw it out? These super coils per se, but to keep it open, Mr. Topoisomerase is the negative supercoiling. Now, once this thing does separate, you got these things on the outside, I should have said them earlier, that keep it unwound, okay? These things, these little, these small proteins keep it unwound, and what are they called? Those are gonna be the single-stranded binding proteins. They keep it unwound, okay? But topoisomerase makes the nick and also does the negative supercoiling. Helicase, we said, separates it. The single-stranded binding proteins keep it, kind of keep it stabilized. Now, once it's out, it keeps it stabilized. The RNA primer, well, he's the guy who initiates the synthesis, right? He initiates the synthesis. Where did he get his power from? He got his power from the energy from the primase, who is DNA-dependent RNA polymerase, given the hydroxyl group. And then this guy here, you remember how we said that you got these little fragments right here? You got these little fragments. Now, after they're made, this leading one's nice and continuous, but these guys down here, well, they need to be stuck together, right? Who sticks them together? Ligase. Ligase is the glue that puts these Okazaki fragments together. Who is Mr. DNA-dependent RNA polymerase? Well, he's the guy that provided, that's the primase, who is DNA-dependent RNA polymerase, gives them the energy for RNA primer. Who are Okazaki fragments? Well, these are on the lagging strand, which is moving away from the replication fork. But again, they're in the 5' to 3' direction, but they're just the fragments, and they're not the continuous. The polymerase 1, now we haven't talked about him. We talked about polymerase 3, right? Polymerase 3 is the guy that lays down the track, right? He's the, he's the guy kind of doing all the work. He also proofreads, right? He has the exonuclease activity or ability because he's proofreading on the go. We'll talk about later on endonuclease activity, which occurs when you can jump in the middle of something. But he's just kind of proofreading as it goes along, so they call it exonuclease activity. Now, DNA polymerase 1, for your purposes, his job is to remove RNA primer, okay, and replace with DNA, right? Remember how we said this RNA primer, which is kind of the guy who's down here just waiting to say, hey, Mr. Polymerase 3, get on your way now, start working. He's RNA, so the job of polymerase 1 is to remove the RNA and replace with DNA. Simple as that, okay? That's his job. This is his job in this world, polymerase 1. You take the RNA and replace it with DNA. Removes the RNA primer and replaces with DNA. And of course, polymerase 3, like we said, he's the guy that lays down the track and does proofreading as such. All right, now that was a lot. But all it is just a story, guys. You tell the story a couple times and you got it, because this is the only way they can ask these questions in biochem when it comes to DNA replication, and it's not that bad. Now back to this picture, which I'm going to tell you, this picture is a little bit flipped from where I wrote it in this direction. But as you can see, this guy looks like what? He looks like a helicase. This guy that's kind of negatively supercoiling, kind of keeping it stable in front of the helicase is going to be topoisomerase. But anyways, this question reads, a diagram explaining DNA replication is shown below. Which of the following would be representative of the function of ligase? Now we said ligase is the glue, okay? But it's the glue of the Okazaki fragments. So we can see, well, let's just look at this. The Okazaki, the Okazaki fragments, we have to identify which one's the leading and lagging strand, right? And if we look at this, if we follow this 5 to 3, okay, so the bottom one is making it in the 5' to 3' direction, right? Well, we like 5' to 3', so this must be a leading strand, and it just happens to be on the bottom. So don't let them fool you, okay? They could put them on top or bottom, we don't care. You got to be looking to see which one goes from 5' to 3'. And since it's 5' to 3' on the one that they're creating, This has got to be the leading strand because it's moving towards the replication fork, okay, towards the replication fork. So this is the lagging strand because they're making it in the 5' and 3'. So in theory, they're making it in these directions. So there should be little chunks of, or little non-continuous patterns that they're going to stick together. And remember, ligase is the glue that binds the Okazaki fragments. So if we were to label these, let's just look at this. Okay, A, as you can see, this one kind of is on both the top and the bottom, or whatever you want to call it. So this is going to be our polymerase 3 more specifically, right? Because he's kind of going to lay in the track, and he's also the one proofreading. B, okay, it looks like B kind of combines these. He was due for a little gap there, and he's solidified that gap. So B could be ligase. Okay, he looks like he's the glue. C, okay, just for our purposes, C, I'm just going to let you know, C is that RNA primer, and he should be on kind of top and bottom. But the RNA primer is just going to be that guy that says, again, hey, Mr. Polymerase, you go. I got my energy from primase. I got who was a DNA-dependent RNA polymerase. So C was actually the RNA primer. D, okay, is identifying these fragments. So, of course, these are the Okazaki fragments. This guy is, again, polymerase 3. These little guys out here, which kind of help keep it uncoiled and stabilize it, these are our single-stranded binding proteins. E is going to be helicase, and then this guy is going to be a topoisomerase. So the guy who's the ligase is the guy doing the glue. It's going to be answer choice B. Okay? Tell the story, guys. You got to tell the story. Last question here reads, research has shown that DNA replication requires the creation of 2 strands of DNA, both in the 5' and the 3' to 5' directions. Which of the following statements is true in regards to these 2 strands? So it's in the directions, and so, but you remember, you always have to create it in the 5' to 3'. So basically, this question is just going to read, out of all the stuff we just learned, which of the statements is actually true? Is it DNA polymerase 1 requires a 3-hydroxyl group from the enzyme primase, which is DNA-dependent RNA polymerase? I don't know about that, because remember we said the only role of DNA polymerase in this world is to do what? It's to remove the RNA primer and replace it with DNA. Now, DNA polymerase 3 requires a 3-hydroxyl group from the enzyme primase, and I also said it was the RNA primer also. I guess I should be interchangeable too. RNA primer requires the 3-hydroxyl group. And I could say the DNA polymerase, and say, to me, they're kind of one and the same for the most part. But what we do know is it's not DNA polymerase 1, okay? It's just not DNA polymerase 1. B, the leading strand moves away from the replication fork. Now, when we talked about that, the leading strand, again, I got to know which direction we're going in. But if it's fragmented on the bottom, because I'm going 3' to 5', 5 to 3, therefore it's 3 up here and 5 there. So the leading strand is moving away. No, the leading strand is going to be moving towards the replication fork. Okay, so it should be towards. So it's definitely not B. Is it C? DNA polymerase 1 removes RNA primers and replaces with DNA. Well, I like it. Let's just keep going. DNA primer is responsible for proofreading the Okazaki fragments. No, it's not. DNA primer is not responsible for proofreading. It's DNA polymerase 3. And is it E, tightly coiled DNA are most active? No, remember, tightly coiled DNA are going to be the inactive. They would be less active, okay? It's the loosely coiled are more active. So the only answer on this, so again, they're just saying, do you know, can you talk about this? Can you tell that story and know which one's true? And the correct answer on this one's going to be C. DNA polymerase 1 removes RNA primer and replaces with DNA. So guys, I know it seems like a lot and it's a mess, but if you can tell that story, I think you can, look through all the terminology, but just understand what's the leading strand, what's the lagging strand. There's only a couple differences between them. And if you get that, and if you can understand that, you'll nail it when it comes to biochem. So hope you liked the video, guys.